Hi, I saw the two hosts ready for the meeting already. So how about you guys? Everything fine?
Yeah.
Ready to go?
Yeah.
Yes. So I think since we have a moderator here, so how about you two just go ahead and try to give us the process the meeting for us. How about that? Are you or we just go ahead and start it?
I think the moderator will guide us, Prof. J.
Okay,
Okay.
that'll be good.
Who's
So,
moderator? Nisa? Oh, Nisa and Gita. Oh, okay. Please, Nisa and Gita will guide us. They as a moderator today. Okay, please, Nisa and Gita.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Honorable Professor Xu Tianchen, as speaker from NKUST and Head of Research Center for Agroenvironmental Testing and Inspection Center, Professor Dr. Mutin Sinurjadi, MSc, as Dean of the Faculty of Mathematics and Natural Sciences and Head of Research Center for Detection of Pathogenic Bacteria.
David Yang and Jason as PhD candidate and speaker from NKUST, Jefferson Dikran SSE, and Tiara Fariza as speaker from UNJ.
Lecturers from UNJ and NKUST, and all students from UNJ and NKUST.
Welcome to all honorable attendees at COIC Touring between Universitas Negeri Jakarta and National Coaching University of Science and Technology on Tuesday, November 14, 2023.
Let us to introduce ourselves. My name is Anissa Fitrianti.
My name is Gusti Anggita. We are a master of ceremony that will guide this event today.
In the first session, let's hear a speech from Professor Xu Tianchen as Head of Research Center for Agroenvironmental Testing and Inspection Center. We welcome to Professor Xu Tianchen. Professor, can you help my voice?
okay, sure. Can you put my slides on? The one says opening. I think that would be easier for the audience to understand what I'm talking about if they have some visual.
Yeah, sure.
How about the voice? Is the voice clear?
Yeah, it's clear.
It's okay?
It's okay.
Okay, so
Okay.
First of
Okay,
all,
we can help to share the PowerPoint for you.
Okay, can you? Okay, good. Okay, here you go. First of all, I want to say hello to everyone here, Professor Modi and your research group and your students too, and also my group and my students to join this wonderful lecture about a couple of subjects we will talk about later. Okay, so it's always been very nice to see the friend from far away. So I think I want to make a short introduction. Myself, my name is Chitian Chen. I am a professor from the Department of Safety, Health, and Environmental Engineering at the National Kaohsiung University of Science and Technology in Kaohsiung, Taiwan. Also, I am a head of a center. I will make some introduction later on today. Okay, so the first question everybody might ask, why we are here today, okay? But I think to me, it's a very creative way that we actually combine two groups, Professor Muti research group and my own group together, and to see if there has anything we can work together. Okay, you won't feel alone every time you have a friend by your side, okay? So I think that's a good thing to work together for research group. And then I personally have contact a couple of research group in different countries, and I enjoy that. And I hope this will be another group that we can work together in a very joyful way. Okay, so actually, in this kind of lecturing or project cooperation, it's a Taiwan national policy. Okay, we have a national policy called New Southbound Policy. We kind of devote some kind of resources and then maybe some interaction between research group or industry or business people. And we hope that between countries, we can make any kinds of cooperation in the future. So that's why I think today, this is just a lecture, but it's a good time for two of our group to know each other. Okay, I think there are many opportunities between our groups. Okay, I think Professor Moody and I will give everybody here in this room right now background information about this group first. Okay, and then we will have a couple talks follows. I think that's a very nice dedicated research data information you can have in today's lecture. And I also want to remind you, this is a 2 weeks lecture. Okay, so that means next week, same time, we have the same similar content talking about research in both groups. And we also set up another time we will talk privately with Professor Moody's group for research. Okay, so it's a multi-front cooperation, and we hope that we can brainstorm us a little bit and then find out what can be done even better. Okay, can I go next slide, please? Okay, so just take the privilege to introduce about today's lecture for everybody. I think everyone maybe even waiting for certain kinds of chance to explore yourself. Okay, and based on the cooperation here in this group, I think the timing is here and the time is now. And then your join in here is also an honor for our groups to present this lecture to you. And as I told you earlier, that Taiwan's new southbound policy, they are aimed for economical cooperation, talent exchange, and resource sharing and regional connection. So a lecture may not be a right one for all of these tasks, but it's a beginning. Okay, so here I think we got the talent, people, the resources, and if we cooperate, and we should be the winner for everyone. Okay, and then we will talk about in this lecture mainly 3 subjects overall: environment, problem, and then nanotechnology and biotechnology. Okay, so I expect this may be just a lecture for now, but we've been talking with Professor Moody about signing MOU or MOA agreements, and maybe we can have a student exchange program and research collaboration on joint project, etc. Okay, and I will talk about that a little bit more later today, about maybe in Taiwan we provide some kind of opportunity to recruit students all over the world to come to Taiwan, and we can learn and study together. Okay, so next slide, please. So I will kind of just take a couple minutes to talk about the program in these 2 weeks. Today is the first week, and then we will mainly talk about each organization a little bit. Okay, so I think Professor Moody is very kind. She will talk about her laboratory and her group in a pathogen bacteria detection. It's very famous. I think it's a very nice research for food foodborne disease or foodborne toxin. Okay, so I think that's the topic worth of listening. And also, I will talk about my group, okay, and then my center and my school, a little bit about Taiwan to you, whoever want to learn more about Taiwan. And then follow that, we have two research talk today. One is led by Professor Moody about her research on foodborne pathogen disease detection. And then the other one is presented by one of my PhD students, David Young, and then he was talking about use of certain kind of nature condensed liquid for bacteria and mosquito inhibition. Okay, and then we will have some research group meeting after the class. Next slide, please. And for the next week, okay, we have more on talks coming up. Okay, and then two is from my group talking about the soil remediation using biotechnology, and then two is from Professor Moody's group. Okay, from Listeria monocytogen detection research. Okay, and I hope we both these two sections we have a Q&A section. We hope we can gather your question and then your idea, and then we can have some interaction or some feedback from you, and then maybe we can answer some of your questions. Okay, that's pretty much the 2 weeks program. So I come to my last slide, and then I will transfer the opening to Professor Moody. Next, please. Okay. Hopefully, you can enjoy the class. Okay, and also find something interesting here to you. Okay, and if you interesting in, I think, either Professor Moody's group or my group, and you have any question, we are more than happy to answer you. Okay, so in my last slide, so I will transfer this opening section to Professor Moody to say some words to the audience. Okay, thank you.
Okay, thank you for Professor Shitian.
Okay, in the next session, let's hear a speech from Professor Dr. Ningsih Nurjadi, MSc, as a Dean of the Faculty of Mathematics and Natural Science and Head of Research Center for Detection of Pathogenic Bacteria. So, we welcome to Professor Dr. Ningsih Nurjadi, MSc. This screen is yours, Professor.
Thank you, Gita and Nisa. Good morning, Prof. Chen, and good morning, all of our students here. Here we mix together between Taiwan student and Universitas Negeri Jakarta student. It's very lovely to have class together with all of you. First of all, I would like to say thank you to Prof. Chen because his idea is very nice to have this co-lecture class between us in UNJ and for your student in Taiwan. This is the firstly we have a mixed class together. So this is the new experience for us. I wish in this collaboration we can have many activity, not only for this lecture, but also maybe we can, as Professor Chen taught us, this is the start collaboration. And then after that, we can have another activity like a conference together, or in 2024, we will invite Prof. Chen to our conference, annual conference SMIC in Indonesia. I wish Prof. Chen can come to Indonesia and give some lecture here. So
No problem.
Thank you. Secondly, I would like to also appreciate for all my student and research group in Salmonella team here who prepare all the facilities so we can we can have class together. Thank you, and I wish this class, not only in this opportunity, we can go further. Maybe the student from Taiwan can come to Indonesia, and then we already sent 3 of our students to Taiwan. Maybe next year, more of students can come to your laboratory and to university. So the both collaboration is more bigger, and we have produced some product or some idea, more innovation in further. Thank you, Prof. Chen. And then as a dean, I also appreciate to your institution because we can have more collaboration, not only with our group, maybe for all the member in our university, because we have 8 faculty and 1 graduate school. Maybe we can send our student not only in my faculty, maybe you can send your student in graduate school in UNJ. Okay, thank you. I
Thank you.
will give back this session to the moderator. Thank you. Assalamualaikum warahmatullahi wabarakatuh and good morning.
Okay, and I would like to add that we would like to invite Professor Modi maybe sometime to join us in Taiwan. Okay.
Thank you. Thank you.
Thank you very much.
Okay, so thank you, Professor Dr. Mufini Sinurjadi, MSE, for your speech.
Furthermore, there will be a group photo session. Attendees are invited to turn on the camera, and please allow the team to help take the picture together. Okay, maybe I will have to count. 3, 2, 1. Once again, 3, 2, 1. Thank you to everyone who participated in this photo section.
So we will enter the main event of this session.
There will be a very interesting material session, but before getting into the material session, we will introduce our amazing speakers today, Professor Shi Tian Chen, Professor Dr. Mufini Sinurjadi, MSE, and David Yang. Okay, I will introduce about Professor Shi Tian Chen. Professor Shi Tian Chen are head of Department of Safety, Health and Environmental Engineering, head of Agro-Environmental Testing and Inspection Center. He was studied at National Cheng Kung University for Bachelor Program, Utah State University for Master Program, and PhD at Civil and Environmental Engineering Department, University of Wisconsin-Madison. His research areas are soil or water remediation, biotechnology, catalytic oxidation, statistic modeling, agro-waste recycling, and reuse. And he has selected publication recently.
Okay.
Okay. Oh, okay. And he has selected publication. And for the last but not least, he also got many achieved in the academic field. And then we can the next speaker. Yeah, please wait a minute. None. Okay, the next speaker is Dun Seng Yang, is a PhD candidate at NKUST. His research area about bioremediation, water wastewater treatment, and agro waste recycling. And now he is a research assistant at NKUST and as technical manager at Actual Biotechnology Ltd. Co. He also has many research publications. And he also has many patents and licenses and achieved in many awards.
So let me introduce Professor Dr. Mukti Nurjadi MSI. She is a head of research center for detection of pathogenic bacteria in UNJ. Her research areas about biotechnology, and she was studied at IKIP Jakarta for bachelor program, ITB Bandung for master program, and postdoc at Gießen University, German. And she also has many research publications. So she also has many patent and licenses and many archives in many works.
Okay, no need to linger any longer. Let us welcome our first speaker, Professor Dr. Mukti Nurjadi MSc. This screen is yours.
Okay, Anissa. This will be okay. Yeah. Here I would like share to all of you about our university, our faculty, and our research group. So we start with our university, Universitas Negeri Jakarta. Good morning. I hope we are all healthy and happiness today. Firstly, I would like to thank Prof. Chen who facilitated the co-lecture between National Kaohsiung University of Science and Technology, Taiwan, and the Universitas Negeri Jakarta. To get know us closely, let me introduce the UNJ, then our faculty, Mathematics and Natural Sciences, as well as our research group under the Center for Excellence in Science and Technology, detecting pathogenic bacteria. So we will start with UNJ. Okay, next slide, please. Okay, please. Universitas Negeri Jakarta was founded in 1964 as part of the Faculty of Teacher Training and Education, University of Indonesia, the oldest university in Indonesia. Then, in 1964, UNJ stood alone as the Jakarta Institute of Teacher Training and Education, or we call IKIP Jakarta. Then, starting in 1999, based on the expansion of the mandate of the Ministry of Education and Culture, IKIP Jakarta changed its function to become a university. So now we call that Universitas Negeri Jakarta. Based on the expansion of the mandate, starting with that years, Universitas Negeri Jakarta provide education for students in vocational, bachelor's, master's, and doctoral degree in non-educational science fields. In every faculty, except in Faculty of Education, there are educational and non-educational study program. So next. Next. To be recognized at the global level, UNJ has a vision, mission, and motto. Vision of UNJ is to become a reputable university in Asia, and the mission is to conduct 3 pillars of higher education, which benefits to the humanity. And our motto is enlightening and elevating the nation. Next. Currently, our rector is Professor Dr. Kamarudin MSE. He was re-elected for 2023 until 2027. Next. For the national accreditation, UNJ had rating for our institution is the excellent, the best ranking national accreditation for higher education institution in Indonesia. Next, please. Several undergraduate, master, and doctoral study program at UNJ received international accreditation from NKUST and ASEAN. Next, please. The location of UNJ is in the capital city of Indonesia, namely Jakarta. Here, our university. Our main campus is in the east of Jakarta region. And next, please. We have 8 faculty. Okay, we have 8 faculty and 1 postgraduate school. One of them is our faculty, Mathematics and Natural Sciences. Okay, next, please. At NIPA UNJ, there are 14 study program, namely Mathematics Education Bachelor and Master, and then Physics Education Bachelor and Master, Biology Education Bachelor and Master, Chemistry Education Bachelor and Master, and then Computational Science Bachelor, Mathematics, Physics, Biology, Chemistry, and Statistics in Bachelor program. In our location, UNJ has 4 main campus. Apart from main campus in Rawamangun, is Jakarta. UNJ has also several locations in UNJ Sport Complex in B campus, and then Faculty of Psychology Education and D campus, and then for the primary school teacher education study program and E campus. Next, please. The facility in UNJ, we have for the learning process, our lecture room, a library, a sports center. Next, please. Dormitory, open theater, laboratory, studio, and we have also hotels in our campus, namely the Naraya Hotel. This hotel is in our campus in Rawamangun, east of Jakarta. Next, please. For the currently, next, please. The Rawamangun campus is under construction. 4 other towers will be built for the several faculty within UNJ with assistance from the Saudi Arabia Fund Development. Hopefully, in the future, more international students will be able to join us. Also from Taiwan. I invite all of you come to UNJ. Next, please. Here the design of our new building, fourth building campus in main campus Rawamangun. Okay. Next, please. Yeah, this is the our new building campus, still in under construction. I wish in the end of in 2024 will be finished. Apart from development at Rawamangun campus, UNJ also preparing to build a new campus in Cikarang area, which will be further development of UNJ as a campus with a legal entity or PTNBH. Okay, next please. Okay, for the achievement, UNJ achievement are achieved both institutionally and by its lecturers and students. As a public service institution, UNJ received an award as the best university providing public information from 2019 until 2022. And then in Indonesia, UNJ is 20 the best university from two. Okay, next. Okay, this is the public award from public relation award. Next, please. For the research and community services, has also produced many innovative products, including products from our Center of Excellence. There are the detecting pathogenic bacteria. Next, please. Not only the students, also not only the lecturer, also the student UNJ have also produced various achievements, both academic and non-academic area. At the world level, UNJ sports students have achieved achievement in the Olympics, ASEAN Games, and various other events. I think we can skip all the achievement of the student because we have a lot from achievement the student. Next, please. Yeah. Okay. If anyone interested becoming a UNJ student, you can visit our website. Here our website penmaba.unj.ac.id. Please come to UNJ. Okay, this is for our university, and then can next to our faculty. Okay. we next to the other purple, yeah. If we you look in slide before, the green color is more contribute in our slide. So for the next slide, this is our faculty color is purple color. FMIPA UNJ has a purple color. For our faculty, we located in Hasim Asari Building, Kampus A, Universitas Negeri Jakarta, Rawamangun Muka, East Jakarta. So next. In our faculty, have 4 leaders here. I'm a dean of Faculty of Mathematics and Natural Sciences, and then we have 3 vice dean. Dr. Esmar Budi is Vice Dean of Academic Affairs. Dr. Sudarwanto SE is Vice Dean of Finance and Finance. And then Dr. Diana Vivanti is Vice Dean for Student and Collaboration Affairs. Okay, next. Our faculty vision in 2030 become a faculty that excel and has competitiveness in the field of mathematics and natural sciences and mathematics and natural sciences education at Asia level based on the faith and criteria. Next, please. And this is our mission. The last is establish and develop collaboration with various institutions, both at home and abroad. Next, please. This is our objective. Next. This is our motto. Our faculty have a motto in Indonesia, we call it Satu. Satu is one. Satu is like acronym for synergy, active, integrated, and excellent. Next, please. This is our organizational structure. We have 14 study program between education and non-education program, and then bachelor and master program. All the activities in our faculty, not only for this lecture, but also for the staff, academic staff, we also have like some activity in our faculty. Next, please. This is achievement of our student. And then we have one of adjunct professor from UTM. I invite also Prof. Chen as adjunct professor in our faculty. Okay, next.
Thank you.
This is a product for collaboration between UTM and IBD. We celebrate the success collaboration and for the 50 joint publication in 2018 until 2020. So I hope also we can have more activity and publication with all of you from Taiwan. Next. For the international community services and international student mobility, we also have several activity for students. This is one of the activity in Taiwan. I think Prof. Chen know better for this activity. And then for our video profile, you can scan here to see on YouTube because it's make some several time to see this. Okay, I will skip and then you can share this. For the accreditation in our faculty, no 6 of 14 study program has accredited by ASEAN, and then I wish in these years 6 others program also can accredited by ASEAN. Okay, next, please. For the facility, we have several laboratory. This is some sample for mathematics laboratory and physics laboratory, and then chemistry laboratory, biology laboratory. This is in our building in Hasim Ashari building. The other facility we have is like for class, library, and others facility who can lecture and student can activity together. So this is our faculty, and then I will next for describe to all of you is our center, research center for detection of pathogenic bacteria, and this is for particularly our group work together. Okay, for this group, for our group, or research center for PUI pendeteksi bakteri pathogen, or research center for detection pathogenic bacteria is driven by energy and passion to give back to society through life science research for a better future. We believe that our product detection kit will give access to every scientist to make the diagnostic faster, better, and more cost efficiently. For this PUI, I head of a research center for detection of pathogenic bacteria. Next, please. Our vision is to become a center of excellence in developing technology for detecting pathogenic bacteria in the fields of biotechnology, health, and health to encourage national independence. Next, please. This is our mission, and then this is our objective. First is producing a rapid kit for detection of food pathogenic bacteria and other sample from various resources based on genomic approach that are fast, sensitive, specific, and accurate for the pathogenic bacteria. And the second is collaborate with various academic and industrial partners in downstreaming research product that can be used to solve problem and improve people's quality of life. This is the overview our research center. I think next, please. The urgency of our research center is, as you know, every human being needs a food in order to grow and survive. The primary requirement that must be met is that the food be of high quality and nutritional value. But microorganism can infect people through contaminated food or drink, included bacteria, viruses, fungi, and a number of other parasites. Based on this background, we have an idea how to make faster, sensitive, and specific for detection bacteria kit. So we grow our research group from this background. Next, please. For the solving the problem, as I told you before, we can make like create some product for us developing a rapid detection kit foodborne pathogen using a real-time PCR method, because we make it this like based on the genomic approach. Our product have like I wish our product have have a characteristic like high specificity, high sensitivity, provide test result in a short time, can be done simply and cost efficient. Next. No, we have 12 products. There are currently 12 PCR products for the detection. If we have in our lab only PCR conventional or gradient PCR, we can use our variant product. For example, we provide for the detection used for the conventional and real and gradient product. But we also provide 12 product if we have like a real-time PCR. So no, our group have like next please. 24 variant product for several bacteria. And this, if you see in the side of this picture, this is one of our product. We call that master diagnostic for kind of bacteria. If you can select the Salmonella typhi, for example, we can use master diagnostic of Salmonella typhi, and then we have two kind of variant here. Variant for PCR conventional and gradient PCR, and then the others product is for real-time PCR. Okay, next slide. For the characteristic our product, we already designed the following item are included: the master diagnostic foodborne pathogen detection kit. We have like 5 tube in every sachet or every product. We have the positive control with the blue tube, and then negative control with the color of pink. Master mix foodborne pathogen, the yellow. And then nuclear free water, green, and the specific primer is red. So if you see our product, we design in a 5 tube, yeah, so we can use it easily. Okay, next. This is the composition for PCR sample. So the if we use this product, we can make like a user friendly. So the user or the technician can only take 1 microliter for the test sample, 12.5 microliter for the master mix and the primer, and then nuclear free water. So we already make this product is user friendly, so more easy to use it our product. So this is the composition for the real-time PCR. Next, please. For the patent, I have already 12 yeah, 11. 11 patented in the granted, and then the other patent, next please, is still ongoing process. We have also 8 in ongoing process. Okay, next please. This is for the books. Next. This is for the IPR. Until 2023, we have 48 IPR. Okay, next please. This is our research member. All of them is very, my student is very innovative, very helpful, and then it's very, can collaborate together. So we invite you to our lab so we can see all of them. Okay, next please. This is our publication. Okay, next. This is our partners here. We collaborate with several academic institution and industry. This is the picture in the left, yeah, right. In the right, for me is collaboration with a forensic laboratory center, Puslapor Polri, and then in the underside here, collaborate with the industry, Kimia Pharma, one of the biggest pharmacy industry in Indonesia, who will produce our product. Next, we also collaborate with other institution, for example, with PT Sinergi Indo Mitra Pratama, who facilitate us to use the several instruments from their institution, and other we collaborate also with PT Trinisah Ersa Pratama to provide for providing instrument to support the work in our laboratory. And then we also collaborate with Pusat Kedokteran and Kesehatan or Pusdokes Polri, was carried out for developing food poisoning detection kit and send the internship students. And this is the list of institutions that contribute with our center for detecting pathogenic bacteria. Okay. This is our small facility in our laboratory. Maybe if compare with your lab, Prof. Chen, is very different. We have only small laboratory, but we have a high motivation to have more publication and product.
Much,
okay. Thank you for your attention, and apologize me if it's take a long time to give more information for all of you. Thank you. Assalamualaikum warahmatullahi wabarakatuh. I give back to moderator.
So thank you very much, Professor Dr. Muftiningsih Nurjadi, MSc, for your material. And now we welcome to our second speaker. That is Professor Sintin Chen. Can you hear my voice, Professor?
Yes, I can. Yes.
Okay, thank you. So this turn is yours.
Okay, can you put my slides on again?
Okay, Professor.
Okay, I think the slides are loading, but at the same time, I will kind of have few comments from to Professor Mukti's speech. It's very impressive. Okay, you have a very nice facility and nice research too. Even have a new building in that school. Okay, it's just too bad. I'm too old to go to your school now. Okay, if I'm younger, maybe I kind of apply your school to study there. Okay, so but I think it's a very lucky student. Lucky of your student can study in such an organization. Okay, your school actually I think it's kind of established. Even I hasn't born. Okay, 1964. It's a very early time. Okay, I think it's a school with history, and obviously you guys run the school very well. Okay, impressive. Okay, so I think it's my turn to talk about myself and my laboratory and my center, and a little bit about my school, and even a little bit about Taiwan. Okay, I would like to say thank you to Professor Mukti again. It's very detailed information about the school and facility. And then as for me, I'm a lazy man, so I didn't really prepare that much slide. Okay, I I do have certain kinds of information I want to deliver today, and I hope that the joint students can maybe get some information from there. And then if you want to have more detailed information about my laboratory, my center, and my school, you are always to come to the website, and I think it's easy to use the website to looking for information. Okay, so I kind of summarize some information, maybe particular is useful for the audience today. Okay, so that's what I want to talk about here. Okay, next slide, please. I think based on earlier Anisa's introduction, you kind of know me a little bit already, but I think it's a very nice opportunity actually to talk about yourself. Okay, talk about myself to the audience. We didn't really do that in the class, but I think today is lecturing, and then I have this chance to say something about myself to the audience. I'm honored. Okay, as Anissa just said, I graduated from Taiwan University, and then after that I went to the United States for my master and the PhD degree. Okay, and then after I'm mostly working on actually the chemistry, oxidation, and also the biological technology, okay, in treating the environmental pollutants. Okay, and after that I got a fellowship from the Rotary Club, and I went to Japan for 1 year and learning the manipulation of the DNA. So I learned quite a few DNA technology from Japan's professor. The professor right now is in Tohoku University, Hanada Sensei, and actually I sent one of my students under his supervision right now. That's who is the speaker later, David Young. Okay, so actually he is connected with us from Japan to talk a little bit about his research. Okay, and so the lecture is, you can see that the most lecture I taught is about biology, biochemistry, molecular biology, and also a little bit about nanotechnology, and something like, I think something related to bio stuff. Okay, and I was the head of a center right now, but I also had quite a few administrative job, part-time job for school. Okay, so in our laboratory, we've been training the entrepreneur from the engineering students. So we hope that students, based on their research, they have certain products, and then they can promote the product and become an entrepreneur someday. That's why I think I kind of play around with the students. Actually, in our group, we go out and then to attend competition, and we won quite good awards from time to time. Okay, and as for my research area, it's basically the soil and water remediation, biotechnology, catalytic oxidation, and recently, it's a little bit about the algal waste recycling and reuse. Okay, and for my publication and then the research, I think because it's too not really that good compared to Professor Muti, so I hide everything away. Okay, but if you want to find my research, actually you can always type in my name and for Google Scholar, and you can see the publication there. Okay, next slide, please. So this is about my center. Actually, we call it the Eco Environment Testing and Inspection Center. It's kind of established under the College of Engineering, and the main purpose actually to set up this center is to help out the farmers, because a lot of farmers along the campus area, they are kind of a certain concern about the food security. Okay, it's the same problem, the food poisoning and then maybe pesticide problem. So they ask the university can set up a certain kind of organization, help them out to maybe take some measurement about certain residue of pesticide or other metals, heavy metals problem. Okay, that's why we set up this center. And since the center is set up in the educational organization, so we also has a couple of service for the neighborhood farmers. Actually, in our center, we let students and to take the soil sample from different farmland, okay, and then we test the characteristics of the soil for the farmer and to give them some suggestion how can they process the soil for their planting plants. And totally we take, I think in my memory, it's about more than 160 sites of soil around the farmland soil in our neighborhood. Okay, so we have a big database for the soil characteristics around our school. And also in our laboratory, we also ask a student to participate in a certain research program. So we actually kind of have a survey or monitoring wetland area for more than, I think, almost 10 years from now. So every year we go to that wetland 4 times. Okay, so every season we take sample from the wetland and to see the water quality is good, and then is there anything about the nutrition or any problem about the water in the wetland area. Okay, and also to monitor the function of the wetland area if they are working properly, something like that. So I think it's a long-term project funded by the Kaohsiung City government. Okay, so other than that, actually we run a more program actually related to the audience today. So I want to talk about more program related to the student more. So next slide, please. So in our center, actually we create certain outreach program for the international students. Okay, so from different organizations of Taiwan government. This is the new program actually established by the National Science and Technology Council. Okay, it's the #1 science and technology organization in Taiwan. They set up a program called the International Internship Pilot Program. Okay, and just started last year. Okay, and it's so hard. So they postponed for, I think, for 300 vacancy, and they use up they use up all the vacancy in about a month. Okay, so the program now is kind of hold for this year, but they will reboot this program next year. Okay, so the aim of the IIPP program, as they call it, they expect some of the interns who join the program from the foreign country will engage in advanced study or start a career in Taiwan. So actually, we are a place. We are in Taiwan. We do need some main force or we need some human resources to join us to work together. So that's why they kind of set up this program, and then they can stay in here for 1 to 3 months. Okay, and last year they opened for 300 vacancies, and then to apply, you need to have a host organization. So on the picture at the left, right, on your right, okay, you can see that our university, National Kaohsiung University of Science and Technology, we already go there and say, yes, we want some guy to come here and work with us. Okay, so we have 3 professors actually set up the demand for the internship. Okay, and I am one of them. As you can see, I'm one of the mentors. Okay, so to apply for that, you need to use the IIPP website, and then you need to prepare a transcript, and then autobiography, and a letter of recommendation. That's it. Okay, and then you go to that website, and then looking for the mentor you like. Okay, and you send application, something like that. Okay, and then the IIPP program, they will pay the intern, I mean the candidate, 3,000 NT dollar, 30,000 NT dollars, each month. Okay, and for the maximum 3 months. Okay, so that's pretty much about the program. So it's kind of a match. You need to find your mentor, and then you apply for it. Okay, if that is a match, then the organization is more than happy to host you to stay in Taiwan for 3 months. But look at this, the small rectangular on the right, that it says that the program, the vacancy is taken already. So they will reopen the vacancy on the December 18th this year. So if you are interested in this program, I think the time to come to Taiwan is kind of next year. Okay, so that just let you know. So if you want to know more information, you are more than welcome to. We have a link here down below, so you can take a look about it. Next slide, please. And the other program that our center set up is also for an outreach program for the international student. We call it a TEEP program. It's Taiwan Experience Education Program. Okay, it's set up by the Ministry of Education, Taiwan government. And the objective of this program is to encourage the international student to participate in a short-term professional internship project. Okay, so work with a certain professor. So we applied a project from the Ministry of Education. They give us certain quota or certain vacancy to recruit the international student to come to the university. And to apply, it's kind of simple. It's just a 6-step. Okay, something like you search for the program, contact the program director, and then submit your application, something like that. Okay, and this program actually is started at 2015, but due to the COVID pandemic, so it's kind of pending for 2 or 3 years. That's why in this year, the 2023, our center actually restart the program, and we do recruit a couple students to come over to our university. So next, please. Okay, so here is the actual student actually start by our laboratory, and they work on certain project with our laboratory and my student too. Okay, so we had totally 4 students come to visit our laboratory this year. And I have to say, 3 of the 4 is from your place, Professor Moody. And I'm honored. Okay, so they did very nice job. I'm impressed. So 2 topics was studied. Okay, one is cauliflower planting soil rejuvenation in Chiaotou district. That's an area close to our university. And the other is use the lake algae for searching valuable substance and energy production. Okay, and this is I had some photo to show some activity that students did in our group. Okay, actually they work very hard in the laboratory. We just show the dying time, but I think they stay more time in the laboratory. Okay, and 4 students from your side, your university, Professor Moody, and then the other student, Jody, he's from Thailand. Okay, from RMUTT University. Okay, I think it's a quite famous university in Thailand as well. Okay, thank you for their contribution in our laboratory. Thank you, Nemo. Okay, and next. Okay, so the center actually, what a couple of professors in the center, we scheduling to do is, as I mentioned, outreach to the international society. Okay, we want to get touch with the different university because of the academic. Okay, so we focus on the connection with different university. That's why I send my PhD student to different university. I sent one student actually to the University of Texas at Austin. Okay, he is the one of a speaker in next week. And I also send David Yang, the speaker, later to the Tohoku University. Okay, so that's why we have a plan actually. We call it the International Youth Agro and Environmental Watch Program, and then we kind of recruiting the member and join our line group in this program. Okay, so we hopefully we will have the international interaction of the worldwide youth to join the program and development of the topic-oriented learning lecture, and then use the online group to discuss the issue about agriculture and environment. Okay, so anyone actually, we are open for next, please. Actually, this program right now we are focused on the Southeast Asia region. Okay, next slide, please. So anyone in this lecture, if you are interested in this program, you are welcome to kind of just join in using the line group with us. Okay, and so I attached a barcode. You can scan and join the line group. Okay, we had about, I think, 30 or 40 members for now. Okay, so next slide. So let's pretty much put about the center part. So the center, we're working on the agriculture and environmental issue. And then also we have the international outreach program. And for about the university I'm in is, we call it National Kaohsiung University of Science and Technology. Okay, actually you can see the whole Taiwan on the right of your, on the screen. And then the university I'm in is NKUST, is in the southern part of Taiwan. Okay, we also have 5 campuses, but I didn't write the name here. So it's 5 campuses, but all around Kaohsiung City. Okay, so right now we have 13 colleges, and we have about nearly 30,000 students. Okay, and 1,620 faculty members in the university. Actually, if you count the student numbers, we are the second biggest university in Taiwan. Okay, so it's a huge university with some multidisciplinary expertise in this university. So I think that's a good place for research. And we also have a graduate school, and we have about 5,000 students in graduate school right now. So the university actually is a very, I think it's the #1 university actually for technology-based university in southern Taiwan. Okay, so and we are very proud that our student actually is kind of favored by the industry. So they hire our student in a very top priority. So I think that's good for the university. Next, please. Next slide, please. And I will say something about the campus I'm in. I'm in the we call it first campus. Okay, and this is a very a natural view campus. But I think compared with your school, I think in Indonesia, I think I saw more natural resources. But here, this place is actually build up this university out of nothing. This is farmland at first, and everything was removed and then rebuilt and then set up this university. So we do have certain kind of design for the university and pretty much a beautiful view in this university. If you have time, Professor Moody, visit our university, then I can guide you personally to our university. Okay, so if you want to see more about this university, please, they have a link about my department. And currently, we have 22 faculty in our department and 400 undergraduate students and more than about 100 graduate students in our department only. Okay, so next slide, please. Okay, and if anyone, okay, just for example, you want to study at the NKUST, okay, actually we do have a program, a nice program to recruiting international students to join us, but I think time is short. I don't want to spend too much time here. I do put on the link. So if you do interest, you are welcome to kind of scan and see the information by yourself. But I do want to emphasize that we have two admission semester. One is the fall semester, and the other is spring semester. Okay, at this moment, the student admission for spring semester has closed. So if you want to apply for Taiwan graduate study for the fall semester, maybe you have to pay attention from the January next year. Okay, so go up to the online application system and you will get all the information. And I show you my department. We do have the bachelor, master, and doctor degree in our department of Safety, Health, and Environmental Engineering. That's the small rectangular on your right. Okay, and I give you some information. If you wanna know more, you can scan the barcode down below. Okay, next. Okay, then the actually to study in Taiwan is come with the scholarship. Okay, and then we have two major scholarships. One is funded by the Ministry of Education. We call it elite scholarship program. It's funded by the government. Okay, and the other one is actually funded by the NKUST alone. Okay, so you can take either one of them, and they have some numbers kind of tell you how much you expect you can receive from the scholarship. Okay, and also we also give you a kind of estimate how much money you might have to spend to living in Taiwan, especially in Kaohsiung City. Okay, so I think just for your reference, and if you wanna see something more, maybe you can just take a look of the slide. Okay, next. Well, I think time is limited, so I want to put up a last slide to show you that we do also, like Indonesia, a very beautiful scenic point around the island. It's a small island, but it's kind of very unique island. We have very high mountain, and we can drop 30,000 meters right away and to the seashore. So it's very nice scenic point around the island. Okay, but I think for the Anisha, this time is just way too short to stay in Taiwan. So maybe we only show them the rural area like Taipei and Kaohsiung City, but not much time to go to the mountain area. But if you want to spend some time in Taiwan, I think it's maybe worth of kind of coming and seeing by yourself. Okay, so I think I will if Professor Moody and don't mind, maybe we can share this kind of information to students as well. Okay, so for the audience, for the students, if you want my slides, you are welcome. I think it's open for you. If you can actually download it, if that's convenient for Anisha and Gusti to operate this kind of operation, you can share the file to the audience. Okay, So that's pretty much about what I talked in sharing the information. Sorry about the time. I think it's a little bit over now. So I think I will give back to the host. Okay.
Okay, thank you very much for Professor Shetiancha. That's very interesting program for us. Okay, now we will also hear the learning material session from Professor Mukti Sinujari, MSc. So the screen is yours.
Okay, thank you, Anissa and Gita. This I would like to share how we develop our detection kit in our lab. So I think the title is Development of Detection Kit for Foodborne Pathogen Disease by Real-Time PCR to Increase Independent Nation. Why we choose this? Because if we have a look at the background, the foodborne pathogen are diseases caused by food contaminated by bacteria, viruses, fungi, and other microbes. The food poisoning case is often referred to as many party, and then the case is sometimes is the extraordinary case. So it requires the contribution of many parties, including the police team, to solve the case. That's why we also collaborate with the police department from Puslapor Polri. And if we have a look the the report, the case report is increasing every years. From I think this is not the last report, but the food poisoning still increase not only in our country, but also in every country, not only in developed country, but also in developing countries. Based on the data from the Center for Disease Control and Prevention, or CDC, in 2011, there are 31 types of bacteria that cause of poisoning or foodborne pathogen that have been identified. Some of that is E. coli, Salmonella, Staphylococcus, or Listeria in Vibrio. We have also learned about the several methods for the detection method. They have like 5 group of the detection kit. The first is for the culture base, and then the second immunoassay. The third is a PCR based method, biosensor, and the others method. We choose the PCR based method or genomic method because the first reason is we collaborate with the Puslapor. They have like the good facility, but they don't like use it very often and very well. So they can collaborate with us. We discuss together. So we choose the PCR based method for detection kit. As you know, as we know, the ideal bacterial pathogen detection method has 5 characteristics. The first is the high specificity, high sensitivity, short time to result, simple or great operational simplicity, and the cost effectiveness. So based on this information, we conduct on we create some detection kit in our laboratory. We use the real time because this method for detecting or quantifying amplified product based on the cooperation of fluorescent reported dyes or fluorescent signal, along with increasing the proportional of number of product per cycle. So we can have a look this more deeply, and then we can calculate and analyze how much the bacteria is contaminated in our food or in our sample. This is my roadmap research. I do my research for when I take my postgraduate degree, and then until now we still concerned to the genomic and the proteomic developing research. Especially in this information, I would like to share of you is for the how we can create the genomic level in the genomic level for the detection bacteria, especially for pathogen bacteria in food. The purpose of our research is to contribute idea on potential research from our university, Universitas Negeri Jakarta, in collaboration with various partners in building the independence of detection kit for the foodborne pathogen bacteria with a molecular approach from Indonesia. This is the methodology. I take this is the what the flow what we do in our lab. The first is the in silico bacterial genomic analysis, and the second one is the primer design. The third is the primer synthesize, genomic isolation, amplification with the gradient PCR to have like more information about the melting temperature. And after we get the melting temperature, we go to the real-time PCR to make the confirmatory to the specificity and sensitivity test. And then after we got all the information, we make the formula and process validation. And then we also like we do some prototype design, publication patent, and then we develop the model for our test kit. This is the result we got it from this every step. The first step, as I told you before, we do some experiment in a silico way. So we try to find information from the database for this is we get it information from in CBI for the Sanger Center. So we take one of the a part of the gene. If I take this as a sample is the Salmonella typhi. For the Salmonella typhi, we try to learn which is a part of the gene of Salmonella typhi, which is contributes to the pathogenic mechanism. So I take a part of this gene. We call this the Fimbriae gene. We choose the Fimbriae gene or Fimc gene. This gene is one of the function is adherent and colonization factor of Salmonella typhi. So if we learn more deep, this gene is have like 693 base pair. So we choose this part of gene, and then we can select a primer from this gene, and then we get it the some primer. One of this is the design of the result of design primer. We get to pair of design, but I give you some information. This is one of pair of the primer. We have two kind of the primer, reverse and forward the primer. For this primer, because we use the real time, we design the amplicode with length 95 base pair. So with this design primer, we can also have information about the melting temperature. So we can go to the synthesize the primer, and then we use it as a primer to amplify the genomic gene or genomic nucleotide for Salmonella typhi. So for this step, we can conclude it for the 3 steps. The first step until the third step, we can conclude if we have like some several primer, and then we already got some information for we choose the particular gene, so we can conclude it has been successfully designed and synthesized our gene or specific specific primer as the target gene for the detection of foodborne pathogenic bacteria of Salmonella typhi. And then we go further to the next step is for the isolation genomic for the Salmonella typhi as a template, because we not only in silico work, but also in the laboratory work. So we start with the isolation of genomic bacteria with some product. We use this is for the Kiagen. So we have like genomic chromosome from bacteria, and then this is for the characterization. If the product is wrong or no, so we can use it for electrophoresis. If the band upper than here, upper than the ladder 1 kb, so if you have look here, the second column until the 5 column, they have like upper than 100 base pair, 1000 base pair. Oh, sorry, 10,000 base pair. So the second column is the for E. coli genome. For the third is Shigella, and the Salmonella typhi, the 5 in the 5th column. After that, we also analyze the purity of genomic bacteria. So if the purity have like a number 1.8 until 2, is the purity is very nice. So we can use it this genomic as a template to the next step to amplify with the specific primer before we design it. So if we use the specific primer to amplify our genomic, so we wish they can have like a specific band in 39 base pair. So we go to the amplified with the PCR conventional, but we use this with the PCR gradient. So the melting temperature from 60 for the 56 until 61. So the band is more clearly in 60 in the 7th column. So we choose this melting temperature or temperature go further to real-time PCR. For this, we can also have like a conclusion. after we get this result, we have also the conclusion. The size of the amplicon of the target gene of STIV has been successfully validated as a detection tool. The optimum cocktail was found for detection of STV bacteria by gradient PCR. As I told you before, we take the 60 degrees Celsius to amplify with the real-time PCR. So we choose this annealing temperature with the 60 degrees to go to the real-time PCR. And then this is for the real-time PCR. Real-time PCR with 60 degrees Celsius, we got the sigmoid curve. This is the red and orange one in the sample of STIV with 56 nanogram. And then this is for the non-template control or for this process. If we have like a difference between sample and NTC or negative control is more than 10, I see in the literature is fine. So because we have like here big difference between sample and negative control is more than 10, so we can make a conclusion. Successful obtain the target gene amplicons at the temperature of 60 degrees Celsius. And then we go also analysis to the melting curve. If the melting curve have like a one peak, so the and then the produced one is very sharp peak. This is like make us is happy because it's the process is going nice. So for the Salmonella typhi, we have also the we also produce the temperature is like 83 degrees Celsius. So we have two parameters for the detection is like CT or cycle threshold and melting curve. If the CT is like make a nice curve like this, like make a sigmoid curve, and then the melting curve as one peak, we can conclusion that process is going nice. So the optimum cocktail was found for detection of Salmonella typhi with the real-time PCR. After this, we also analyze the specificity for our product to differentiate between bacteria target and the non-target. So in this result, we also can differentiate between Salmonella typhi as a target bacteria and then the others bacteria as a non-target bacteria. And then we also make a sensitivity test. For the sensitivity test, we also got information our product is very sensitive because we can still detect is in a picogram concentration. So if we have looked the if we have a dilution here, we can dilution for 10-fold 7 until 8. And then after we got all the we got the data, we can conclude for this step the primer design can distinguish target and non-target bacteria so that they are a specific one. And the film-stay primer reaction mixture or cocktail and design RT-PCR collision could detect target bacteria at the small amount, the picogram level, so their words is highly sensitive. And then we also make experiment for the artificial contaminated. We take the Salmonella typhi to food, and then we incubate the Salmonella typhi with the food, and then we isolate also all the bacteria and the and then food without the bacteria, and then we can run together as a control positive. If you have a look here, the Salmonella typhi non-dilute and egg. This is means we don't make a dilution for this sample, and that we make a dilution until 10-fold 6 minus 6. They still have can detect this Salmonella in the food. So we also can detect in pure culture and in the contaminated food. And we also try to select some food and then make a test for this, for the Salmonella typhi, and then for the other Salmonella typhi. And then we have also conclusion, then our product can still use it for the differentiated or can detect the bacteria in food and it culture pure culture. So after we make like optimize and optimize not only in cocktail or in some condition, we try to make like a prototype. This is the first our prototype, and then the new product specification we also create, because we learn some information, and then we change our product with the new type like this. So as I told you before, we already have 12 master diagnostic for several detection kits for Salmonella typhi, E. coli, Shigella, Salmonella typhimurium, until the last is the Vibrio alginolyticus. So now in our lab still going to experiment to validate and efficacy test in some food product, for example, for the meat and for some various food. So for this, especially for the Salmonella typhi, we have 4 publication and 1 patent produced, and then the result of this research can the conclusion. Our conclusion is like the result of this research can contribute to the scientific repertoire handling forensic cases, as well as the implementation of the result at the partner institution and the wider community to increase the independence of a nation. This is our grant for this years. We have 3 of kind of the grant from our university for our ministry, and then for the Riset Innovation Indonesia Maju Badan Riset Inovasi Nasional, or BRIN. This is all my collaboration for student. We call Salmonella team. We have for all the Salmonella team here. Okay, there are Salmonella team for 2018 until 2020. And this collaborate partner with our team, Pusla 4, Kimia Farma, PT Sinergi Indo Mitra Pratama, and thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Well, very nice. Very, very nice. May I ask a short question?
Okay, please.
Yeah, just very short. The time is kind of constrained, but I just wondering, because you mentioned that you make some kits already, right? The product kits for detection some pathogen. I just wondering, what is the expiration date for that kit you made? For example, how long if I buy, for example, if I buy the kit, okay, is any expiration date for the kit?
No, we didn't produce the kit and then go to society because we have like a MOU with the industry, with the Kimia Pharma industry, the big pharmacy industry in Indonesia to produce that. So I follow the rule. They will produce it in their industry. So like we sign the MOU first. After that, go to the society. So no, we didn't produce that. I wish in the coming years or in the end of the year, the MOU sign is finished. After that, we go to the production.
Okay, thank you, man, Professor Modi. Thank you.
Thank you.
So thank you very much, Professor Muktin Nisi, for your presentation. And in the next session, we will hear the learning material session from David Yang. For David Yang, can you hear my voice?
Yeah. Hello, everyone.
Okay, thank you. So this time is yours, David Yang.
Yeah, I can share my morning time.
Wow, it looks very cool in Tofuk University.
Yeah.
Oh.
Okay.
Now it's 11 degrees.
11 degree. Okay. Wow.
You are still in Japan?
Yes.
Oh, okay.
It's a good thing for online lecturing. I mean, yeah, we have the audience from all over the place. So that's good.
Hi there, my name is Vincent Yang. I'm from National Kaohsiung University of Science and Technology. I am going to here present my study is use of smoke and condensed liquids from inhibiting bacteria and the mosquito larvae. And they have many permanent growth fruits like date, guava, and pomelo around my university in Yanchao District. Fruit trees produce many branches each growth, and too many branches need to be pruned. So many western branches are produced. They are also speak on some western branches, which can damage farmers when put on the farmland, and these western branches will be open while the combustion, but now is prohibited by the law. So farmers don't know what to do to treatment these so many branches. And western fruit tree branches in Yanchao disease have 5,000 tons per year. And here can see guava 1 year needs to treatment twice times, and the date once times, pomelo once times. So have produce western fruit trees branches each year. And they have 3 ways to treat with branches. First one is open field building, but is prohibited, and the second is compost, but it needs use long time to fermentation. And the third is smoldering. It can produce biochar and the smoker condensed liquid and return to farmland to use. And smoldering is a slow formless form of combustion and have several advantages. Like a collective process with possible pollution control and this carbon dioxide production and no extra energy required during its process. And their reference show different trees to make liquids have different antibacterial and kill insect function. Like first one have higher temperature, have stronger inhibition activities to bacterial. And this study want to use this liquid to treatment mosquito. So we need know mosquito growth history. The first one is larva, and about 5 to 8 days can become pupa, and the mosquito, and the reproduction to egg, and the egg need 3 to 4 days can become larva. And mosquito cases and dengue fever, Zika fever, and the yellow fever. And he have harmful human body, have like high fever and the muscle and bone, chest, and the others. And here is Taiwan every year have dengue fevers case, and this year have more people take dengue fever, and this is very dangerous because his mortality rate high as 20%. And how to treat a mosquito, we can see a lot of time is used chemical agents to kill, but these agents can damage our body too. So the objective of this study, including the following. First one is the goal of this study was to manufacture the SCL of 3 branches from date, pomelo, and the guava. And we analyze ingredients of 3 SCL and test SCL battery restore and the mosquito larvicidal effect. And the second is the basic characteristic of each HCl ingredient change over time and different temperature. And how to take HCl? We put 500 gram wood chip in the reaction tank and the smoldering, and the smoke will collected in the condensation tube and can take HCl in here, and this is HCl. And after smoldering is over, there will be biochar in the reaction tank here. And we need analysis HCl. So metal analysis, we take 1 milliliter sample and 1 milliliter nitric acid and 3 milliliter hydrochloride and put in the COD reactor heating 50 degree 1 hour and 95 degree 2 hours, and feed it and use SPAS to analyze metals. And ingredient analysis, we use 5 milliliter S10 and use sanitizer to extraction. And we have take extraction sample and internal standard to GC mass analysis ingredient. And bactericidal test, we take 0.2 milliliter bacteria liquid on agar plate and distribute this liquid, put filter paper and HCl on the agar and put in the oven 35 degrees for 2 days, and then we can measure the diameter of growth inhibition. And the mosquito larvicidal, we take 10 milliliter various concentration of HCl in the bottle, and the various concentration of HCl bottle. Every bottle put 10 larvae and record the number of larvae dead over time, like this picture. And properties of 3-fruit-tree HCl, we can see the HCl pH is low, is 2.6 to 2.9, and no analysis heavy metal in this liquid. And major ingredient of 3 SCL, we can see the date. He have high abundance than the other two SCL and have high phenol-Likers compounds more than pomelo and the guava. And we test battery residual effect, and the SCL 2.5 concentration is no battery residual effect. The 3 SCL is same, and in 10% date SCL and the pomelo SCL is high 1.44 and 1.30 times, then 75% ethanol. So the higher the SCL concentration have the great battery residual effect. And we put 3 SCL in 4 degree 0 to 180 day and 25 degree 14 to 180 days, and heat 80 degree 30 minutes. And we can see the 80 degree 30 minutes, the date is drop 25% and the pomelo is 32% and the guava is 32% abundance. And in inhibition zone area, the date is drop 29% and 43%, 51%. And we can see this picture, the battery residual effect was most likely proportional to the content of the SCL. And this is major compounds and abundance lost this. Then in 25 degree 180 days is lost than 20%. So the SCO can long-term preservation. And in mosquito larvae straw effect, we use different concentration. And in 10% SCO and the 5% SCO can faster kill mosquito larvae in 2 or 3 hours. It's very fast, and that is faster than pomelo and guava. And maybe because mosquito and because larva become to mosquito, it become to pupa need 5 to 8 days. So maybe we can use 0.6 to 5% concentration to kill larva in 100 to 60 hours to 100 hours. and we want to see different pH can affect to mosquito larvae straw if. So this bank use DD water and in pH 3 and pH 11, the kill mosquito larva time is very long, around 117 hours, but in 10% SCL can in pH 3 and pH 11 can kill larva in 1 to 3 hours, 2 to 7 and 1 to 4 hours. Can kill 100% larva. And because larva grow in the water needs oxygen, so we want to know if the SCL reduce diesel red oxygen effect in water, use 10% SCL in the water, and sampling time is 0 to 3 hours, and we can see the three kind of SCL can affect this solvent oxygen in the water. So the larva are killed because SCL's phenolic compounds is soluble. And this study conclusion follow. The first one is the SCL is acid and no heavy metal, and the most compound is phenolic compound, and 3 SCL have similar organic compounds. And the great abundance of the SCL liquids have the high battery residual effect, and the highly positive correlation is it between the SCL abundance and the battery residual effect, and ambient temperature, the depth and the polymer abundance lost less than 20% in 118 days. So we can long-term preservation, and we use 10% SCL can kill the larva in 2 to 3 hours. And this is I have for today. Thank you for your listening.
Okay, thank you very much, Davidian, for your presentation. And so there will be a question and answer session, and the questioners are invited to use the feature to raise hand. So you want to ask, keep raising hand.
Well, I want to add a little bit is that maybe you guys have some experience about dengue virus as well. Actually, this very year in Taiwan, we have very serious problem about dengue virus. So how to prevent the growth of larvae in the environment is kind of a very important task for us. So maybe I think the dengue virus is a common problem in the Southeast Asia region. So if anyone has any comments, maybe you can share with us. Okay. So if not, maybe okay, I think that the following is the Q&A section, right? But since the time, I think is kind of a little bit over now. So maybe how about that? If the students still have question want to ask about today's talk, you might want to leave the message on the board, okay? And then maybe the hosts can transfer the question or their question to the speakers, okay? And then if not, maybe it's about time to we can end this section for today, and then we are expect to see you guys next week. How about that, Gusti? So I see some student here on the board. So Izu Ting. Okay. Okay. I see some familiar faces here. May I ask, which grade is the biotechnology class? Anisha, do you know?
Yes, Prof. Sorry, can you
I
repeat?
mean that the class of biotechnology, the student in which grade? Is in a freshman or is a sophomore, junior, senior? Class?
Third for 4 year.
It's a senior course.
Yes.
Okay, so that means everybody is about to graduate. Is that right?
Yeah, that's right, Prof.
Oh, okay. So I hope that some information I share today, you can think about it. Actually, I think it's a good opportunity to maybe contact or outreach to the foreign country. Especially, I think Asia country is not really far away from Indonesia. So think about it. And please, the hosts, you can share my slides to the audience, to the class. It's fine with me. Okay.
Yeah, okay, Prof. But maybe we will give the last opportunities to other students. If
Mhm.
you want to ask, you can raise your hand. Is anybody here? Yeah. Okay. Here's Valia Rahma. You can say the question.
Okay, thank you for moderator for the time I can asking my questions to Mr. Yang. I am Valia Rahma from Universitas Negeri Jakarta. Maybe I want to asking you about the using of the liquid smoke to kill mosquito in the experiment was concluded to be effective. However, is there an impact on the environment? Can you explain for apa? Can you explain more for me? Maybe just thank you.
Is that clear for the question?
Sorry, you can whisper again.
Maybe I want to asking you about the using of the liquid smoke to kill mosquito in the experiment was concluded to be effective. However, is there an impact on the environment? Can you explain me for more, please?
We have tested different conclusion like pH or dissolved oxygen, and we don't analyze have any metal in this liquid, so I think maybe it's organic compound like phenol or the other ingredients have anti-bactericidal or mosquito larva residual effect. So I think maybe it's phenol liquid compound. So, but this part we need, maybe we can buy some phenolic compound to try to kill larvae. Thank you.
Well, I think it's a great question. I want to add something more about it. I think we do have certain concern about the environmental effect to the small condensed liquid. Okay, and then because actually that's why we at first we analyzed the ingredient using the GC mass. So we actually detect each single ingredient in that solution. And we do observe a lot of phenolic compound. We do need some, the toxic ingredient to kill the bacteria and the mosquito. But for us, okay, the smoke, actually, if we release into the environment, they are even more toxic into the air, okay? But since we already condensed that liquid into the smoke into liquid, actually we kind of prevent the air pollution. Okay, if we just burn it in open space, then we will create some kind of smoke, and then we do have air pollution, and we have a PM 2.5 air pollution problem, something like that. Okay, so just in terms, we kind of condense it into a liquid, and we kind of monitor the toxic compound. We didn't see very toxic one like the dioxin or other compound. And since it's a agricultural plant, they don't have sulfur, so we don't produce sulfur dioxide, something like that. So relatively, that liquid, I think if we use it properly, they can be degraded in the environment in a short time. But we just don't have time to show that yet. Maybe your question is good indication that we might be able to do something like that. We trace those phenolic. Once they hit the ground, how long they will last in the environment. And if we can prove that, maybe we can show you that they will be degraded, I think, in a very short time in the environment. Okay.
Okay, thank you, Valia, for the question, and Professor Shitian Chen and Dun Seng for the answer. Yeah. Okay, we also open the question for maybe UNJ and NKUST students who want to ask about UNJ or NKUST, or maybe the programs available to our presenters today. Is there any questions so far? Or Professor, do you want to say something or ask to our student? Okay.
I think that's about, we can discuss that. I think that's have some time to think it over. And then since we will meet again next week, okay? So maybe we can discuss more questions next week. And then by the time we have more time, okay?
Okay, maybe we have one questioners. Yeah, is it Dean? You can say the question, please.
Okay, is my voice clear, Miss Anissa?
Yeah,
Okay,
Yes, I
clear.
can hear you good. Okay.
okay, okay. Thank you for the time for asking. Actually for NKUST and UN Jakarta. And actually for Chen, thank you for having me. I'm from Group Line of International Agro Environment. My name is Izzuddin. I'm from Indonesia and actually in Yogyakarta. A graduate student from environmental engineering. I just communicate with you about 2 or 3 weeks ago. And my question is about my research background with about the wastewater treatment. And I want to ask about the how wastewater face the future. And actually, we face the problem in Indonesia with water treatment and with disposal. It's use conventional technology conventional. We just throw the waste and we just throw the wastewater in nature without any technology. So is it possible in future, and actually in developed countries like in Indonesia, we have some research, more details in our research? Is it possible to make our country be more tips in research? It's maybe that's it. Can you get my point? Pardon for my question.
Okay, is that a question for me, right?
Yeah, naturally for you, Mr. Chen, because
I will try to answer your question from couple direction. Okay.
Yeah.
To me, it seems number one, wastewater is a problem. It is a problem. Okay, and it is a problem everywhere. Okay, as long as we have any kind of activity, we have any production, then we have the wastewater problem. Okay, so but to be specific, there are many kinds of wastewater. Okay, it's organic polluted wastewater, it's metal polluted wastewater, it's pathogen polluted wastewater, many kinds of pollution in the wastewater. So if you're asking about innovative or new technology to treat the wastewater, yes, we do. And yes, we do have many very exciting technology available nowadays. Okay, we have more advanced biotechnology method, and we have more advanced catalytic oxidation method. to treat those pollutants in the water. Okay, but I think since I just said earlier that there are many kinds of wastewater problems. Okay, so I think that will depend on case by case, which kind of pollution you are talking about and what kind of technology we might be used to treat that wastewater. Trust me, wastewater treatment is a very steady demand all over the world. Okay, everybody need to deal with the wastewater problem. So I think that's a question we deserve. We put more time to investigate. Okay.
Okay, Prof. Chen, maybe this is a related question. Maybe Miss Anissa, can you translate into English? Maybe I need some translator. And actually, because that question, jadi ketika saya riset, Prof, jadi saya berpikir bahwa ketika saya riset, apakah saya bisa memberikan kontribusi kepada negeri saya. Padahal di negeri saya itu dalam arti di teknologi dalam sekitar saya itu masih jauh memerlukan teknologi yang saya buat. Miss Anissa, can you translate it maybe?
Yeah. He say when he doing the research, he want to think about can he help to the country about the result, his result. Yeah.
Can you say it
Something like
again?
that,
Sorry.
Prof. Yeah, he said student said, can he make a contribution to the country while technology is very minimal? Yeah.
I think sure, why not? Okay, especially for if you don't really use a lot of technology yet. Okay, so maybe I think doing something is worth of trying because okay, sometimes I want to say is that technology is not really that expensive, especially you're talking about the newly developed technology. They always focus on biological treatment, for example, and then they use the local material to create the environment for bacteria growth and for bacteria to treat the pollutants. So it just takes some time to study the problem, and then we can propose some method. Actually, I have a student from Bangladesh, and they have certain problem about the food wastewater treatment. Okay, but I think that's a good thing because in your country, maybe you have certain kind of problem like that. But actually, we in Taiwan, for example, two of my students actually is from food production company. So they have the same kind of problem. Okay, so we can just learn from the current way people treating the food production, manufacturing wastewater. Okay, so we can kind of modify a little bit maybe or do some a small modification, and hopefully they will suit your needs in your country or your case, something like that. So to me, use of technology is always worth of trying because otherwise the pollution will continue, and then eventually the environment will kind of broke because the environment cannot take so much burden eventually. So treating wastewater, I think, to me, is very important task for everyone. Actually, for right now, people kind of looking for problem in air pollution and water pollution. That's two most kind of close related to our daily life. So people pay attention to the pollution very closely. So I think that if you have exact wastewater problem, yeah, I'm more than happy to kind of discuss with you more. Is that okay?
Okay,
Okay.
thank you, Mr. Prof. Chen,
Yeah, thank you, Is.
for motivation.
Okay, thank you very much for the questioner and our speakers today.
Is there any more questions so far from the participant? Okay, if there are no questions, we are all ready to end this session. So thank you for attending the co-touring between Universitas Negeri Jakarta and National Kaohsiung University of Science and Technology today. We also have a second opportunities to talk about our research in the next week. And I'm Gustinggita Putri, and my partner is Anissa Fitrianti. We are as a moderator and master of ceremony today, and we want to say goodbye. So thank you very much for all the participants. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, take the opportunity to thank Gusti and Anisha. Nice to host this section for us.
Yes, Professor.
And thank you for you for joining. Thank you
Thank you
for
very
you all.
much.
Bye-bye.
Thank you very much, Professor.
Bye.
Yeah, bye-bye. Have a good day, everyone.